students. Today I will discuss on different mechanisms involved in the life cycle of Bacteriophage lambda and how they regulate the expression of Bacteriophage lambda genome, and also how Bacteriophage lambda is used for various techniques. Bacteriophage lambda belongs to the family Cephophyridae, and it has an icosahedral head of 55 nanometer diameter and a non-contractile tail of about 150 nanometer with a thin tail fiber attached at the end of the non-contractile tail. And K12 strain of E. coli is a very common host for lambda bacteriophage. Lambda bacteriophage is a temperate phage. That means it can go for either lytic cycle or lysogenic cycle depending on several factors. If it goes for lytic cycle, then it will replicate its genome inside the E. coli cell. It will the structural proteins will be formed. The structural proteins will form the protein coat, outside coat, and the newly replicated genomes will enter there, and new viruses will be formed and will be released from the host. But if lambda follows lysogenic cycle, then it will incorporate its genome into the E. coli chromosome, and incorporated lambda genome is called a prophage. The prophage state is very stable and it replicates along with the E. coli chromosome. The genome of lambda is about 48.5 kilobase pair long. It is a double-stranded linear DNA molecule with 2 GC-rich 12 nucleotide long single-stranded overhang at both the ends, and they are complementary to each other, so they can form base pairing, and thus these two sites are called cos sites. And Friedrich Sanger first determined complete sequence of nucleotide sequence of DNA molecule, lambda DNA molecule, in 1979. If we look into the lambda genome, all the genes are arranged in order of their function, okay, and very easily lambda genome can switch over from lytic cycle to lysogenic cycle and lysogenic cycle to lytic cycle, and this observation led Jacob and Monod to propose the famous operon model of gene regulation in prokaryotes. And now we know that the actual mechanism is much more complex than that. Indeed, it has an operon system of regulation, and 3 operons exist in the lambda genome. The left operon codes for the genes responsible for recombination integration of the lambda genome into the host chromosome, that is, left operon is responsible for lysogenic cycle. The right operon is responsible for replication mainly, and the late operon carries all the genes necessary to produce the structural proteins, that is for lytic cycle. Now immediately after entering into the E. coli cell, what happens? Circularization of the lambda DNA and immediate transcription of some genes, of 2 genes. This is called immediate early transcription. Transcription starts from 2 promoters PR and PL and terminates at 2 termination sites TR1 and TL respectively. These 2 short transcripts give rise to 2 proteins, N and Cro. N protein acts as an anti-terminator. Now we will study the mechanism of anti-termination. There is a site called NUT site, NUT for N utilization site. NUT site is present before the termination site. For each of the 2 genes, before TR1 and TL1, 2 NUT sites are present. NUT site is composed of 2 sequences, 2 specific sequences called box A and box B. The box B region forms a stem loop structure as soon as it is transcribed into an mRNA. Clear? So when this region is transcribed into an mRNA, in the mRNA box B will form a stem loop structure, and N protein will bind to the stem loop structure, and N protein secondary structure will be changed. Now the changed secondary structure of N protein will interact with RNA polymerase, okay, and it needs help from 4 other host cell proteins. They are called NAS proteins for N utilization proteins. Host protein, NAS protein A, E, B, G plus the changed in protein and RNA polymerase together form a complex called anti-termination complex. This complex now overlooks the termination site TR1 TL. Also, it will overlook any other termination site coming into its way. So, once N protein is bound to a RNA polymerase molecule, that RNA polymerase molecule will proceed through all the termination sites. Clear? This is how the transcription anti-termination occurs. Now, when N protein will anti-terminate the transcription will proceed in the rightward direction as well as in the leftward direction. From the rightward direction, all these proteins Cro, C2, O, P, Q will be produced. From the leftward direction, N, C3 will be produced. Now, Q protein again acts as an anti-terminator. How it acts? When Q protein is responsible for promoting transcription from the promoter PR prime, which is a promoter for lead genes. All the lead genes, that is the genes encoding structural proteins, are encoded in this region. Now, when RNA polymerase, E. coli RNA polymerase starts transcription from PR prime promoter, it will continue transcription up to 16 or 16 nucleotides. Okay, then it stops because it confuses this site, the pause site, with the promoter site. Okay, that is the pause site has a sequence similarity with the promoter site. The sigma factor is still with the RNA polymerase. Now, Q protein comes and binds to the halted RNA polymerase. It replaces the sigma factor and pushes the RNA polymerase from the pause site. Again, RNA polymerase continues transcription, and as long as RNA polymerase transcribes, Q protein stays with it and it helps to overlook all the termination sites. Thus, transcription of all the late genes is possible with Q on board. Now, C2 protein activates transcription from three promoters, PRE, PI and PAQ. Transcription from PI will lead to the synthesis of an enzyme integrase. Transcription from PAQ will lead to the synthesis of a small transcript which is antisense to qmRNA. And transcription from PRE will lead to the synthesis of C1 and antisense Cro. Here comes another beautiful example which is present in lambda phage, that is antisense control of regulation of gene expression. Here you look, the PAQ promoter is present in the Q gene but at the opposite strand. Okay. So, the transcript produced from PAQ after transcription is complementary to the qmRNA. So, as soon as these two transcripts are produced, they will bind together and it will stop the expression of qmRNA. That means no Q protein will be produced, and in the absence of Q protein, no transcription from PR prime promoter is possible. So, it will stop the lytic cycle of the lambda bacteriophage. Similarly, transcription from PRE promoter will lead to the synthesis of a transcript which will carry antisense of Cro mRNA plus C1 protein. C1 protein is required to establish lysogenic cycle, Cro protein is required to establish lytic cycle. Now, since a single transcript is carrying message for synthesis of C1 protein and message for anti-termination, anti-Cro protein, it will at a time will stop the translation of Cro mRNA and it will lead to the synthesis of C1 protein. Thus, it will help to establish lysogenic cycle. Now, the big decision, lytic or lysogenic, is taken by the lambda genome. Now, it depends on the operator regions, two operator regions present in the lambda genome, operator rightward and operator leftward. Each operator region is again divided into three small regions, OR1, OR2, OR3, similarly OL1, OL2, OL3, and each operator region, small operator region, is of 17 base pair long and they are almost similar in their sequence. And if we look into the affinity, and both Cro protein and C1 protein has affinity towards the operator region, but the orientation is different. Cro has maximum affinity for OR3 region, then for OR2 region, and it has least affinity for OR1. Similarly, affinity of Cro is maximum for OL3, next OL2, and least for OL1. And affinity of lambda repressor or C1 protein is maximum for the OR1 site, then for OR2 site, and least affinity for OR3 site. Similarly, OL1, OL2, OL3. So when Cro protein concentration is more than C1 protein, Cro will bind to OR3 region and it will block the PRM promoter, but it will allow its own synthesis from the PR region, PR promoter. And when more Cro repressor is present, it will form dimer with the existing Cro, and when more Cro protein is present, it will completely inhibit its own synthesis. Okay. And when lambda repressor is present more, it will bind to OR1 first, preventing transcription from PR, but it will allow transcription from PRM. PRM is always active, but transcription goes on at a very basal level from PRM. Now when OR2 region is blocked by lambda repressor, it will interact with the RNA polymerase, and now transcription from PRM will be activated. More C1 proteins will be produced. At a very high concentration, it will block the OR3 region also, stopping the or blocking the PRM region and stopping the transcription from PRM. Here comes another beautiful mechanism called autoregulation or feedback inhibition of the repressor protein. So at a low concentration of C1, it will first bind to OR1, OL1 region, then it will attract another C1 dimer to bind to OR2 and OL2 region. And when C1 protein concentration is very high, that is, it happens actually after integration of the lambda genome into the bacterial chromosome, then another dimer of C1 will bind to the OR3 and OL3 region. Okay. Operators and Cro proteins bind to the operator rightward and leftward in the similar fashion. Now, C1 protein has a dimerization domain as well as a tetramerization domain. Okay. So, the dimer present at the OR3 and OL3 region, they are without a partner. Okay. So, to form a tetramer, the DNA bends and forms this structure, and the dimers present at OR1, OR2 and OL1, OR2, they also form and they also bind and form the octamer. This is called the feedback inhibition, since C1 protein blocks its own synthesis. And again, when C1 concentration will be low, since it has least affinity for OR3 region, this region will be free, and again PRM will be free. So RNA polymerase will again continue transcription from PRM. Thus, C1 protein maintains lysogeny within a very narrow range. Okay. And presence of C1 in a cell will block the operator regions of another lambda genome. If another lambda genome infects the same cell having a prophage, okay, then its infection will be, its genome will be blocked by the C1 protein. This is called the immunity to superinfection. This is called the immunity region, since all the operator regions are present here. Now, the lambda genome integration into the host chromosome occurs by recombination process called site-specific recombination. It is called site-specific because the lambda genome integrates at a very specific site present on the E. coli chromosome between the gal and bio-operon. Okay, and the site is also specific in the lambda genome also. It is called the attachment P site. Attachment P site on the lambda genome and attachment B site on the E. coli genome, and integration occurs by an enzyme called integrase. The recombination occurs via formation of holiday junction and exchange and rejoining of the DNA molecules to form two recombinant DNA molecules. The attachment sites present in bacterial chromosome as well as in the bacteriophage lambda genome, they have a sequence like this. Okay, 15 base pair common sequence flanked by dissimilar sequences B prime in the bacterial chromosome, P prime in the lambda chromosome. And this bracketed 7 base pair sequence is actually taking part in the during the site specific recombination. Now, if we look into the map of the lambda genome before and after integration into the E. coli chromosome, then we will see in the vegetative map, that means the map that is the DNA, that is the map in the DNA when the DNA is present in the phage head. At one end A gene is present, on the other end R gene is present. But after entry into the E. coli chromosome, if we look into the map, it has IN gene at one end and J gene on the other end. That means though the relative order of the genes is still the same, the ends are not same. Okay, that means the vegetative map and the prophage map, they are called, they are cyclically permuted. Very good, cyclically permuted. Okay, and Alan Campbell first proposed it in 1962. He proposed that after first proposed that after infection the lambda DNA circularized through the two cohesive ends and the attachment site is different from the cohesive end. Okay, so circular form integrates into the chromosome, bacterial chromosome, by opening a site other than the cos site. Now, it is very stable. Now, the lysogen thus formed is very stable. The prophage, the genome integrates and stays as a prophage until the DNA, until the host cell DNA gets damaged, okay, by UV ray or by some chemical mutagen, anything, okay. And when DNA damage of the host cell occurs, then what happens? The prophage comes out from the integrated stage. This is called induction. And induction requires two enzymes, integrase and excisionase, whereas integration requires only the integrase enzyme. Okay, and the way lambda phage ensures that only integrase is synthesized during integration, but both integrase and excisionase are synthesized during excision is a very regulated by a very beautiful mechanism called retroregulation. Retroregulation is a regulation when a sequence downstream of a gene regulates the expression of that gene. Okay. So, what happens after infection? From PL promoter, transcription starts from the PL promoter, and since it has a NUT sequence, N protein will help RNA polymerase to overlook all the termination sequences. So, this termination sequence will be overlooked, okay, and transcription of this region will produce the stem loop structure, and this stem loop structure is a cleavage site of RNase 3 enzyme. RNase 3 cleaves here, then RNase 2 chops the DNA. Okay. So, though both INT and XIS genes are present on the same transcript, they are not translated into proteins. So, after infection, these two proteins are not synthesized from the PL promoter. But lambda requires the integrase enzyme. Then how it is produced? It is produced from another promoter called PI promoter, which is present within the XIS gene. Okay, C2 activates transcription from the PI promoter. So, the integrase enzyme only will be produced after infection, and it will lead to the lysogenic cycle. But after induction or during induction, both the enzymes are required, integrase and excisionase. Then how it will be produced? Now, if we look into the map of the prophage, okay, the attachment site is present here, okay, before the RNase 3 cleavage site. So, during integration, this site is cut, okay, and the genome integrates into the attachment B site. Now, at one end it has INT gene, on the other end it has the RNase 3 cleavage site. They are separated now by the lambda genome, okay. So, again from PL transcription will start, but since no RNase3 cleavage site is present here, it will not be degraded, and this transcript is very stable. Now both the int and XIS, both the proteins, enzymes will be synthesized, and they will help in the excision of the lambda genome from the integrated stage. Excision of lambda genome is again requires site specific recombination, and now the integrase excision is have to recognize a hybrid sequence. But sometimes the excision is not proper. During excision, sometimes by mistake a part of the bacterial genome comes with the viral genome, and a part of the viral genome remains in the bacterial genome. And when this new phage, defective phage, infects another bacterial cell and the genome integrates into its chromosome, then it is getting an extra copy of the bacterial genome. This is called the specialized transduction. Now what happens, and specialized transduction is very rare. It happens 1 in a million of the bacteriophages, and it is called the low frequency transducing bacteriophage lysate. When this low frequency transducing bacteriophage lysate is used to reinfect the gal bacteria, okay, then there is a chance that 2 transductants will be produced. One will be integrated at its normal site, attachment B site. The other one will undergo recombination or crossing over with the homologous region. Thus, 2 lambda genomes will be integrated into a bacterial chromosome, which is called double lysogen or dilysogen. Now, lambda bacteriophages are used as vectors also, cloning vectors. Cloning vectors are small piece of DNA taken from either virus or plasmid or an eukaryotic cell, and which can accept a foreign DNA. We know that an advantage of lambda vectors are that they can accept quite a large foreign DNA fragment, and several thousand clones can be screened at a time in a petri plate, and very easily they can be stored as lysate, clear lysate at 4 degrees centigrade. Now, as bacteriophage genomes are packaged into the head depending on the cos sites. If we have a DNA having 2 cos sites 50 kb apart, then very easily that DNA can be packaged into the phage head. This is used to generate plasmids. Plasmids containing cos sites can be packaged into the phage head. It is called cosmid, and they are very helpful for in vitro packaging, okay, where we have alpha infected cell extract, that means all the head, tail, everything is there, and we have this cos site attached foreign DNA. Then this DNA will be packaged into the head, and tails will assemble to form the complete bacteriophage. Now this bacteriophage will attack we can use it to attack the desired cell. Now, very recently a new technique has been developed called recombinering or recombination dependent genetic engineering. Okay, it depends on the homologous recombination in E. coli using the recombination enzymes of lambda phage. Okay, in the red operon of lambda phage there are 3 genes, red alpha, red beta and red gamma. Red alpha gene product is an exonuclease which cleaves the 5' from 5' end of a double stranded DNA. The red beta gene product is a single stranded binding protein which helps in the annealing, and red gamma gene product, it blocks or prevents the activity of reg BCD. Okay, that is and they induce a hyper reg condition in E. coli and other bacteria, and they promote the recombination if only 40 base pair similar sequences are present. Okay, thus now very simply we can do the genetic engineering or based on this recombinering technique. Lambdaphage has been used as experimental tool to discover many other things, like Samuel Benzer discovered the or determined the extensive and fine structure of the R2 gene of T4 bacteriophage using the Rex gene of lambda. Also, Werner Arber discovered the restriction and modification system using lambda as a tool. So today we have learned what are the different mechanisms present in the life cycle of lambda, how they regulate the life cycle and expression of the lambda genome, and how can we use lambda for research going on today and for future.